Let me introduce our speaker today. This is Dr. Zachary Ardern, who is a research fellow in evolutionary genomics at the Wellcome Sanger Institute. His scientific interest is the origins, functions, and evolution of new genes in bacteria and viruses. He is interested in his spare time in the historical and philosophical development of biologically informed natural theology, which is integrated with the evolutionary history of life. Thanks, Zachary.
Well, thank you, everyone. I'm really glad to have the chance to share my thoughts with you, and I look forward to your questions. So are there underlying principles of protein evolution reconsidering law and purpose in biology? These are some of the concepts that I'll be introducing today on the left, and some scientific fields on the right. And I'll try and draw some connections between some recent developments in the science on the right and some very old concepts on the left. These are massive areas, and I'll just be touching on some of them in a way that I hope is fruitful. So how can we learn about the past? One way to do this is in archaeology with a class of tools known as stratigraphy. The basic rule being that the older layers of, say, an archaeological dig are deeper, and the shallower layers are younger. The same principles apply not just for archaeology, which was worked out relatively early, and soon after these principles were worked out, they were applied also in geology for much longer time spans. So we can go back hundreds of millions of years using these kinds of broad principles, and we can see a history of the Earth and of life when we dig through the layers in the Earth. So this is stratigraphy in archaeology and geology. But we can also learn about the past through molecules, through molecular sequences. We can infer history by considering patterns in the modern distribution of traits of biological organisms, but also amongst people groups. So for instance, I think a helpful analogy is the evolution of language. So this was worked out back in the 1600s, that there is a relationship between, for instance, different European languages, which don't immediately on first sight look like they're related, but you dig into certain core words, you see similarities, you can actually infer an evolutionary history of language. And some of these same principles actually apply for understanding the evolutionary history of life as well. So the information molecules of life, such as proteins, are a bit like words in different languages. And in similar ways, we can infer histories from many of these words or proteins. So this is a field called phylostratigraphy, or genomic phylostratigraphy, where we can look at genomes. So this is just a very simplistic representation of 4 genomes, or a section of 4 genomes, with a few genes in each. And here I've colored the genes according to whether they're widely shared or only shared by a few species. So here we see that genes A, B, C, and D are relatively old. They're shared across all these 4 species. These might be divergent species. We imagine 4 different mammals, like bats and mice and chimpanzees and humans, for instance. But there's some genes that are only shared with a few in a smaller group of species. And subject to various caveats, we can infer that these genes are probably younger. So we see some genes are very old. They're actually shared across the whole of life. They're shared between bacteria and archaea and eukaryotes like us. There's some genes that are very young. They're restricted to a single species or a single genus. So these genes are illustrated in orange here. And this whole area of study is called phylostratigraphy, or genomic phylostratigraphy, when we use genomes. to infer the history of different traits. Okay, so now I'm going to share some thoughts about how we came to discover this history and how people have thought about it theologically and philosophically. So a first question, which might seem really basic, is does the Earth actually have a linear history? This has been surprisingly controversial. It's something we take for granted when we do stratigraphy or phylostratigraphy, but many different cultures and thinkers around the world through history have said, actually, there isn't a real history to the world because everything doesn't really change that much over time. Everything has always basically been more or less the way that it is today, or everything goes in cycles, where any history is kind of fleeting and then it returns back to the start. So there's no real kind of point in exploring ancient history if everything is just going to cycle forwards back to the same point again. And I'm not an expert in any of these thinkers, so I may have got some of these details wrong, but the basic idea is that many people through history have thought that the world is eternal or eternally cyclical. So for instance, James Hutton says, we find no vestige of a beginning, no prospect of an end. So here's one of the early modern geologists. And this is a theme that I'll come back to a bit more later in exploring the challenge of deep history to a Christian worldview. Many people would think that the discovery of deep time was kind of a victory for atheism or naturalism, whereas I'll argue that actually the linear direction of history, including deep time, fits better with a Christian philosophy or theology of nature. Some of these early people who were atheists or naturalists or deists who believed in an eternal universe actually didn't really believe in deep time in the sense that we do today. So when did we start to discover that the world is really ancient? Maybe earlier than you might think. So at least often in introductory biology lectures, it's kind of implied that it was Charles Darwin or someone about the time of Darwin who discovered the Earth had an ancient history. Actually, this was known about well before Darwin. Robert Hooke, the famous microscopist, acknowledged that there have been many species of creatures in former ages. He even speculated that there might have been new kinds that came into existence, but he probably didn't think major groups of animals there. He probably thought just varieties. And he also realized that these fossils that he was discovering will probably far antedate all the most ancient monuments of the world. So he gives the pyramids as an example. So he's realizing that these fossils are really old, much older than we could imagine with our knowledge of human history. So he recognizes extinction at the top. He recognizes some aspect of biological novelty. And he recognizes a source of information through which we can learn about the history of life. So this is amazing, I think, back in the late 1600s, that he had already kind of starting to put together the basic building blocks of our modern knowledge of Earth history. Okay, so what is the nature of this deep time? What controls it? What kind of processes are happening in it? One of the main emphases in the early 1800s is people were starting to learn that there is a law-governed history to the universe. So sometimes it was proposed that as we discover laws that can explain various features of the world, that might have been seen as supporting atheism or undermining theism. But people like William Whewell, the master of Trinity College, back in the early 1800s, said, actually, this is not correct, that a law-governed history to the universe doesn't have to be seen as atheistic. He says, with regard to the material world, we can perceive that events are brought about not by insulated interpositions of divine power, but by the establishment of general laws. So in other words, his developing picture of the universe is that the way God interacts with it is through laws primarily, not through interventionist action. And very strategically, I'd say Charles Darwin uses this quote at the start of The Origin of Species to implicitly suggest that what he's offering is a law-based account of the history of life to complement the law-based account of the history of, for instance, the solar system, which had been recognized in the kind of decades and maybe century before Darwin. And so this on the left is an illustration of the nebula hypothesis, which had been worked out a few decades before Darwin as the likely origin of the solar system, which was a law-governed process. And this is not too controversial today. Most Christians and others are happy with a law-governed unfolding of the physical universe, but it's more controversial regarding biological universe. So what about that? Well, Charles Babbage, an early founder of mathematics and computing, was also starting to develop ideas of a law-governed unfolding of life around this time. So he said, in like manner, does God impress his creation with laws? God's omnipotence entails he can foretell what causes are needed to bring about the effects he desires. He doesn't need to intervene every time some new cause is required. God is like the inventor of a complex, powerful calculating engine. And a lot of people point out that it's kind of funny that he likens God to the inventor of a computing machine when he's the first inventor of a computing machine. He's clearly seeing himself as doing something godlike. But this idea of a law-governed unfolding of life was in the air before Darwin was writing and around the time. Richard Owen, a little bit before The Origin was published, spoke to the British Association on what was summarized as the axiom of the continuous operation of creative power, or of the ordained becoming of living things. So elsewhere, he talks about secondary laws, which God has put in place to allow the unfolding of the history of life. After The Origin was published, he put it this way: the inductive demonstration of the nature and mode of operation of such secondary continuously operative species-producing force will henceforth be the great aim of the philosophical naturalist. In other words, discovering the secondary laws which God has put in place to allow the development of life, that will be the new focus of biology. Okay, so that was a quick look at some of the history. And what about proteins? Where do they fit in here? Well, a lot has been discovered about proteins recently. There's some people in the room I know who are more experts on proteins than I am, so I'm curious what they think about this. But I'll give a quick summary to what proteins are and the diversity and structure before I think about the evolution of proteins. So there's different levels of protein structure, and most of us will be familiar with this, that we start with the primary sequence of amino acids, and these can fold into small motifs known as secondary structure. And together these form the complex folded structure of a protein. And proteins can operate together in units, in complexes, which together can achieve remarkable things. For instance, the bacterial flagellum is one famous example, and there's many of these molecular machines or complexes in the cells of eukaryotes and bacteria. So there's different levels of protein structure, from just the chain of amino acids to how it folds at the local level, and then a more global level, and how it interacts with other proteins. Now, AlphaFold is a reasonably well-known recent development, which was just awarded the Nobel Prize in chemistry, along with some related work in computational protein design. Just a quick summary: how does AlphaFold work? I think something that many people, including biologists, don't fully appreciate is that the basis of AlphaFold is the co-evolution of amino acids. so that across a chain of amino acids, so if one position in the chain changes, for instance, the blue position that's illustrated there, this will often affect other positions in the chain, and it will be more likely to affect those positions in the chain that actually fold to be close to that first position. So positions that are close in three-dimensional space, even though they might be far along and separated at different ends of the chain, will actually tend to co-evolve together. That if one changes, there'll be a selective pressure to change the other one to compensate for that. And these patterns of co-evolution, when you compare a lot of sequences to each other, can be used to infer which amino acids are most likely next to each other in three-dimensional space. There's also many developments in artificial intelligence that actually make use of this information and refine it. But together, the use of huge amounts of evolutionary information with recent developments in artificial intelligence allow us to infer the structure of proteins. And now there's a few different related methods for doing this. Why is this interesting? Why is this useful for the biologist? Well, in most cases, we can now find the approximate structure of a protein of interest within a few minutes or less. So now this actually changes evolutionary biology, that until a few years ago, we usually were studying sequences, but now we can study structures instead. And structures are a step closer to biological function. And so it's opened a huge amount of potential research into understanding the evolutionary history of life. There's been another tool developed subsequent to AlphaFold called ESMFold, which is a faster tool. It's a large language model. It's a different artificial intelligence approach. And this has been used to infer 700 million proteins, their structure, which allows us to really map the full protein universe in a way that just wasn't possible a few years ago when we had experimental structures for only something like 200,000 proteins. Okay, so what can we start to learn about evolution through these new methods? Another aspect of biology that's not that well understood, including by many biologists, is that some genes are younger than others. So we know that through evolutionary inference that everything can be traced back to some kind of last universal common ancestor, and we can infer the genes that would likely have been present in that ancestor, Luca, the last universal common ancestor. And we can compare those genes to the genes we see in different organisms, and we can try and trace their history. And it turns out that there's many genes that are present in modern day organisms that do not seem to have been present in ancestors going back even a few million years, let alone billions of years. So how can we explain this? There's basically two possible mechanisms. One is just that the sequences have evolved so fast, we can't see evidence of their ancestry. And the second, which is C here, is that actually new protein coding genes arise from non-coding sequences. And there's growing evidence that this is a major source of new genes. So it used to be believed that everything would trace back to the last universal common ancestor, but increasingly we see evidence that there are genuinely new genes arising through evolutionary history from non-coding sequences. And this broadly is the area of work that I'm doing research in. So basically these sequences which used to be non-coding occasionally become coding. They get translated through different mechanisms that we're just starting to understand. Sometimes these non-coding sequences are situated between pre-existing genes, and sometimes they're partially overlapping pre-existing genes. And it's that second one that I've been particularly focused on. And if anyone's interested, there's some papers on that I've worked on. There's different mechanisms by which sequences which used to be non-coding, but were overlapping a coding gene, can evolve into a protein coding gene. This is all kind of quite new, and there's a lot more to learn about how genes really originate. So gene origin is relatively poorly understood, but protein evolution is relatively well understood. So we understand a lot about the evolution of proteins within protein families. With structural comparisons, we can sometimes trace the evolution of proteins even once they no longer show any sequence similarity. So this is just an example that I like, 2 proteins in the virus, SARS-CoV-2, that causes COVID. And if you look at them closely, there are actually some similarities, but if you do a sequence search with BLAST, it'll tell you no significant similarity found. So at the sequence level, you couldn't tell that these things are related, but based on the structure, you could guess that maybe they are. If you look closely, there's kind of this transmembrane domain is basically shared between these 2 proteins. And people have actually compared all the different coronaviruses and related viruses and worked out a plausible evolutionary history to these 2 proteins, which based purely on sequence properties, you wouldn't actually be able to tell are related to each other. So protein structure is conserved much better than protein sequence, and that allows us to infer really ancient history for proteins. Okay, so is protein evolution law-like in some way? Are there long-term trends, or is it purely subject to natural selection, which we might expect would fluctuate as the environment changes? On the standard Darwinian picture, where natural selection dominates, we don't expect long-term trends because the environment is always changing. There is some literature historically on generally long-term trends in evolution. There do seem to be some trends in that things are more complex now than they were billions of years ago, or at least some things are, just putting it very simply. There are various long-term trends that have been suggested that, at least in some lineages, things do seem to get more complex over time. So that's interesting, but what about proteins? There's also recently some work on protein evolution, on some potential long-term trends there. I won't talk about this in a lot of detail because I'll skip to the work that I've been doing recently. But I just wanted to highlight this. This is not completely new. There have been a few papers proposing different long-term trends through evolutionary history in proteins over the course of billions of years. Something that's relevant here when thinking about long-term trends is the fact that convergence seems to be so widespread in evolution. And one of my favorite examples is actually between the bacterial flagellum and a very similar machine called an archaeal flagellum or an archaelum, which is found in archaea, which are quite distinct separate-celled, single-celled organisms. And as far as we can tell, these 2 machines are not evolutionarily related to each other, but they have some remarkable similarities. So they're both rotary motors that are used for propelling the single-celled organisms through their environment. Their protein structures are actually remarkably similar in various ways as well, in terms of the protein monomers that they're made out of and the complexes that they form. So this is a remarkable example of convergent evolution. And there's many of these through life, but so far not many of them have been studied at the protein level in the way that this one has. So what's the potential explanation of this convergence? So there's work by Ard Louis in Oxford, who some of you will be familiar with, who I think is really starting to unpack some of the explanation for why we see some of the convergences that we do. And it's basically that some structures are simpler than others, and simpler structures are basically easier for evolution to find. So this is an illustration of basically complexity, or you can imagine that the left of the x-axis is kind of the more simple structures, and these are the ones that we find more commonly in nature, basically. So we can start to explain the kinds of complexes that proteins will tend to form based on some general principles, like a massive bias towards simplicity in evolution. And inspired by some of this work, I decided to look at whether there might be trends in protein structure itself. And I think there are. So I'll just give a couple of examples. If we look at what are the structures of young genes, of genes that look like they're restricted to a species or a genus, and are filtered carefully in various ways, it turns out they're incredibly simple. They're basically usually just a single alpha helix, or 2 alpha helices. If we look at the older protein domains that are shared much more widely, they're shared across different bacterial phyla, they're much more complicated, and they tend to have not just single helices, but they also have a bunch of beta sheets in some arrangement. So older proteins, it looks like, are much more complex than younger proteins. This is a trend that we find across species. So this was a gram-negative bacterium, but we see the same thing in gram-positive bacteria. And I think this is quite striking and interesting, and actually not what people really expected. People don't expect consistent trends. This is a work in progress, and there's still some work to do here, but basically I see the same trend, and I see it at a more granular level. If you look at genes which are restricted to the species or the genus, they're simpler in the sense that they include more alpha helices than genes which are much older. So basically genes over time seem to be accumulating beta sheets or protein domains do. So this is looking just at the protein domain. So this is the folding part of a protein. I'll make an argument that alpha helices really are simpler to justify my claim. So one reason to think that they're simpler is that if you just attempt to fold random sequences, they're more likely to fold into an alpha helix than a beta sheet. And we see that here, where I've taken sequences from lots of different genomes for different genome compositions, and you always get either 1.5 to 3 times as many helices as you do sheets. Helices also evolve faster than sheets do. And this makes sense if there's kind of more ways to be a helix than there is to be a sheet. That means if you change a helix, it's less likely to change away from a helix into something else. And so we see, this is just preliminary data, but we see on the left that helices have a higher evolutionary rate than sheets. And this has been reported before, but hasn't really been put in an explanatory context like this. And thirdly, I think interesting, but I'm not sure exactly how this fits in, but helices seem to fold faster than sheets do, typically. And this is just some data from a database of protein folding where the helices are in blue, and they always have a higher folding rate than, overall, they have a much higher folding rate than sheets do. So it's been suggested that helices typically take microseconds to milliseconds to fold, whereas sheets take milliseconds to seconds. So this idea that they're simpler and easier to find, both in evolution and biophysically in folding, is interesting, and I think fits with this general progression from simple to complex that we see in protein evolution. Okay, so what can we say about teleology in evolution in light of these things? Broadly, there's 2 approaches to teleology in biology. One is to focus on the adaptedness of creatures to the environment and their exquisite designs. And this is, interestingly, something that William Paley and Richard Dawkins have in common with each other. They're both focused on why species are so well adapted, so well set up to live in a particular environment. So that's one approach to teleology, to thinking that there's some kind of driving force behind that. Another approach is called structuralism. And this is where the focus is not on the function, but on the overall body plan or structure. And this is focused on the underlying orderly plan, which is found across the biological world. So this was broadly the kind of school of thought of Richard Owen, and also perhaps that of Simon Conway-Morris today in Cambridge. So in this talk, I've been inclined towards the second path, though actually the way that these things are related is very subtle and interesting. And someone like Richard Owen was actually very much involved in thinking about both of these. So related to this, that evolution in some sense is law-like, and the history of the universe has some law-like aspects, but there's also a lot of contingency in there. There's a lot of events that seem highly historically contingent, or not law-like, not able to be predicted from first principles. So for instance, the formation of galaxies seems to be a law-like process, but the formation of the moon seems to be a highly historically contingent process. Origin of life, or maybe Paul Rimmer will have thoughts on which one dominates there, whether it's law-like or contingent, but that's a very much an active debate. The origin of, similarly with the origin of animal body plans. But both of these factors are important in evolution. And I just don't want to take away from the fact there is a lot of apparent contingency in evolution as well as law-likeness. So there's some open questions still, many open questions. How much in evolution can natural law or underlying principles account for? Do we need some kind of fine-tuning of these laws? Will any general principles do, or do you need special ones? I strongly think you probably need special ones, but that's an open question. I think it's interesting that the fine-tuning that we're aware of in physics will give us a life-permitting universe, but I don't think it guarantees us a life-evolving universe. I think there's extra requirements to get that, but very much an open question how constraining this requirement is. Okay, so back to the historical question of whether the universe is in some sense directional. An argument that Professor Martin Rudrick makes in this very nice book is that actually there is an analogy between the concept of deep time and the Judeo-Christian concept of a historical process which God is involved in. So he says the strong sense of history gave the Judeo-Christian tradition an underlying structure that's closely analogous to the modern view of the Earth's deep history, as similarly finite and directional. So we don't live in an eternal universe, and we don't live in a universe that's going nowhere, but it does seem to have some directionality, maybe even in the history of protein evolution. If you're interested to learn more about any of these things, these are some books that I highly recommend. I think you've had various of these speakers at the Faraday Institute before. And on that note, I look forward to any questions that you have.
So there's a microphone in the room. There is a question online. Let's start with someone in the room. Yes.
Thank you very much, Zachary. Very, very interesting talk. I was kind of curious about your sort of picture of the simple genes and the complex genes. And one way which you seem to imply that you could explain that is maybe these genes arise de novo, and they tend to have the alpha helices, and then they just get the beta sheets later on. But a picture that you showed was kind of a debate between two different views. One was that everything starts with LUCA and then evolves from there. And then the other is that you get these de novo genes, and it's probably a mix between the two. Yeah. Could this be a sort of argument that there's like a sort of kind of pre-weighted high complexity sort of state, and all of the old genes happen to have that because it's all been kind of pre-weighted to more complex things, and so all the de novo things just can't really get to that state very easily? Or is there good evidence of older de novo genes that also have these sort of beta sheets?
Yes. So after that, I showed a plot, which I didn't explain very well, that I tried to isolate genes that looked like they were de novo originated within the higher groups as well, not just the species specific, but also the family specific. And you do seem to see a consistent trend there. It is hard to be sure that you're getting genes that originated de novo, but I think this kind of trend would be very surprising in the other picture as well. Like, why should the genes that look like they're really young be so simple if actually everything has a really ancient ancestry, which was the usual view of protein evolution until fairly recently. But yeah, there's a lot more to do to really be sure that we're talking about de novo genes here. Yeah.
Can I ask another clarification question while we're on? So someone is asking bacterial versus archaeoflagellum. So you said you don't think they're related. How do you know they're not related?
So it's based on the structure of the individual proteins, and they just come from very different protein families. So this has been studied in some depth, and basically they're just completely separate cellular systems. And there's some looking into the potential ancestry of these systems as well. And they seem to be coming from quite different places.
So like comparing a chimpanzee to a zebra.
Maybe I need to think more carefully about that. But
Ignore that last comment. Yeah, there's another question in.
Yeah, hi. I had a question about I suppose, the language of law.
Yes.
In the sense that we have a legal system. I wonder if law implies a lawgiver, whereas evolutionary constraint, which itself is extremely complicated, doesn't. And you sort of making an argument that a lawgiver is required for this, or that, because evolutionary constraint is really, really complicated, and I wonder if all of this is just evolutionary constraint, as in it's a process plus time. Gives you simple things quickly and complicated things after a long period of time.
Yeah, great. There's a few different questions there. Firstly, that's an interesting philosophical question for the concept of scientific law in general. And I think there's actually good arguments to think that scientific law, certainly historically, came out of a Christian view where there was a lawgiver. And philosophically, it's actually hard to make sense of some of the properties of scientific law. And there's Nancy Cartwright is an atheist philosopher who rejects laws of nature and has a different approach to causation in science because she thinks laws of nature are too theism friendly. And there's various others like that. So there's a kind of growing literature on laws. But I didn't mean it in any loaded sense. I just meant scientifically, there are constraints here that might not seem controversial, but actually within evolutionary biology, it is very controversial because it's quite different to the Darwinian picture, which is that it's random mutation plus natural selection. Evolutionary constraints would be a third factor. And if actually constraints are the dominating factor that give you these long-term trends, that would be distinct. So you might be able to tell a story where you get the same trends through natural selection. So that could still be possible. But some of the things that have recently been discovered in biology, for instance, in Ard Louis' work with RNA, seem to be really pushing more on the law side of things and don't seem to have an explanation through selection. Another way to put this is that the assumption of Darwinian evolution is that mutations are random, but actually if they're massively biased towards a subset, which we call the simple subset, that's actually a different picture of evolution than the Darwinian one. So yeah, you're looking skeptical, but I can suggest some more papers on this, and it really is controversial, the role that constraint might play, because it isn't actually either mutation or natural selection.
Thank you. I have another question online. I should say to those online, please put your questions in the Q&A. I cannot see Q&A and chat at the same time. Please stop using the chat. Put your questions in the Q&A. So we have one online. I've always imagined the arrow of evolution from simple to complex. Your comments on convergence towards simplicity seem to be in tension with this. Can you comment on this?
Yeah, so it's more just where evolution, yeah, I think that's a great question, but the way I'm thinking about it is, where does evolution tend to go? What kind of directions does it tend to go in? And it actually makes sense from some first principles that it would tend to start in the place where it's easiest to start, where you're more likely to hit upon a certain phenotype. So those phenotypes, which are the starting phenotypes, should be more common, and evolution will be biased by what it can start with. This is what we see with de novo genes. I think that de novo genes start where they're most likely to start. And I think that's true for evolution in general. That doesn't prevent it accumulating complexity over time, but it does dictate what's the most likely thing to happen next in the evolutionary process is whatever's most common in the phenotype space around that.
Kind of like a biological Occam's razor.
Yeah. You want to, evolution can only work with what it is given. And if it turns out that what is given is hugely biased, that's actually going to shape the evolutionary trajectory in a way that hasn't really been appreciated until relatively recently.
Let's have one in the room there.
This may be following on the question you just addressed, but I was wondering if you could comment on the relationship between the drive towards simplicity in protein structure and the graph that you showed about a drive toward complexity in organismal, I take it, capability over time. where you had hominids, birds, and then reptiles and whatnot. Do you see these as complementary? Are they dissonant?
Yes.
Are they unrelated?
Yeah. Yes, similar question and a good one. So I don't really see it as a drive towards simplicity. It's more what is the dominating force in the evolutionary process. And I think it is that whatever's, given there's different options, whatever is most common in the phenotype space is the one that's more likely to happen. And I think over time that will actually allow you to accumulate complexity. There's more work to do to tell that story of how that works. But at least you don't start with so what's surprising is that all the possible outcomes aren't equally likely. There's some that are much more likely, and that's going to drive evolution in a particular direction. That's about all I can say.
We'll have another one online. This one includes very loaded language for a biologist. What's your view on insights for the new to nature protein designs? Do they look like young designs?
The new
to nature protein designs. I think that was where you were talking about two ways of proteins, new proteins coming about.
Oh yeah, the de novo one. So they look, so that was my point, I guess, that they do look simple, and they look very different to the old protein. I think that's all I can say on that. Unless they, by new to nature, they mean things that are designed by humans. I could say more about that, but yeah.
I suspect what we're all interested in is how you interpret that with respect to the existence of a creator. I can push you. People would want to jump to conclusions.
Sure. So what I'm saying is that there's a bunch of recent evidence in evolutionary biology that evolution is more law-like than we expected. And this actually brings us back to some pre-Darwinian views like those held by Richard Owen and others who were structuralists. That's very consonant with a picture where there is a God who is a lawgiver. And some aspects of Darwinism, the strict emphasis on randomness, seem less consonant with that. So that's about as much as I can say on that. Yeah.
Okay. Just maybe a simpler question. So how do you differentiate between younger and older genes, and how would you compare that to appearance of mutations?
Yeah, so this was one of the first slides that if a gene is shared by only a few organisms that are closely related to each other, then we can infer that it's younger. So similar to some genetic traits in the human population, if there's a trait that's only found in a very small subpopulation, we can infer that probably wasn't found in the ancestor, but has arisen like a mutation de novo relatively recently. So the same with genes, that if a gene is only found in a certain species of bird, and it's not found in all the closely related species, the most parsimonious inference is that it arose relatively recently. And we can kind of build outwards from there. The larger the group of organisms, the older the gene broadly.
Thank you. Just coming back to something you've already touched on several times, but you talked about a complex dance between contingency and law-like behavior. And I'm wondering if part of the complexity or part of the discussion arises simply the kind of time level that we're looking at these things. In other words, are you standing back and looking at the whole process over a few million years or a few 100,000 years, or are we looking at these initial, these sort of sometimes dramatic contingent of events that do obviously change things a lot? And this is probably a really bad analogy, but I'm just thinking of the evolution of traffic lights around the world. There's a lot of pressure to have traffic lights for them to exist. There's a lot of convergence as you get a lot of traffic coming in different countries. But then there's a lot of contingency at the local level. If you're looking at individual traffic lights, like the ones down the road here, then they vary a lot depending on the environment. And so sort of thinking about that, is there kind of two levels here we're looking at? Is that useful idea, or does it not really fit with what you're saying?
Yeah, so if someone thinks the evidence for structuralism is really clear, then you could definitely see the adaptations as kind of icing on the cake. They're kind of like local level, short, over relatively short time periods, you see contingency, but at the longer level, you see, and I think something like that is true if you do see these really long evolutionary trends that I'm talking about billions of years. If these trends are real, and there's a bunch of evidence that they are, then you have to see the role of adaptation as relatively minor in regards to the properties that we're looking at. There's some properties that might be strongly affected by adaptation. Yeah, contingency, and there's also some contingencies will have massive implications for billions of years of evolution. Like, let's say the origin of the first eukaryotic cell was a massively contingent event. That's massively shaped evolution ever since. So yeah, that's why it's a dance between law-like processes and some important but rare contingent processes.
Hi, thank you for your presentation. I was wondering, perhaps, because you mentioned this idea of perhaps a trend towards simplicity, could it be that perhaps there's some sort of middle ground between the two positions where things that are simple also tend to survive better? So for example, I suppose if this is a good analogy, if you were to overcomplicate a protein structure, it's much harder for a cell to create that protein. There are more points of failure. Whereas if there's a simpler protein structure, there might be an inherent adaptational advantage towards that. Would that be possible?
Yeah, so I think something like this is why it's surprising that there is so much complexity, because you would often think simplicity would be biased. There would be a bias towards simplicity through selective processes. So what I'm trying to argue is it looks like there's non-selective processes. And part of the reason I think that is that the trends are so consistent across time. And I think that's hard to make sense of through a selective process. I might be wrong about that, but yeah, I don't think there's like a massive overall trend towards simplicity through selection, because otherwise we would just see simplicity everywhere.
Thank you.
Can I push you to a more philosophical question? As is the question of beauty in protein structure, which some would think is a sign of something transcendent, maybe imagined as a trace of God in creation.
Yeah, so there's like an interesting relationship between simplicity and beauty. And I think part of the reason protein complexes are beautiful is because they are quite simple. This is like the work of Ardern, that there is this bias towards like multi-member protein rings and such, which are actually much simpler than they could be. The alternate explanation for that will be laws of nature. Then the alternate explanation of laws of nature will be God if you're a theist, and will be unexplained if you're an atheist. So insofar as laws of nature are producing valuable things, I think that is a pointer to God.
I'm wondering if you'd agree that observations of nature that show a combination of law and contingency make good theological sense if we're to understand that God's providence isn't a sort of overruling power, but actually God works in and through nature's unfolding. So it's not either law or contingency, but actually, theologically speaking, both speak of God as creator.
Yeah, I mean, something that I think about there is the analogy to the history of the people of God, which seems to be maybe dominated by contingency. But at the same time, there are some underlying themes that Christians would say come to fruition in Jesus. So there was a long-term plan there. So actually, like the history in the Old Testament, or the Hebrew Bible, I'd say, is very messy and contingent, but still ordained by God. So that's one argument I'd give for those who think that evolution is inherently problematic because it's messy. Actually, human history is messy, and God is still ordaining that. I'm not strongly inclined to a view that gives creation a lot of freedom, because I'm not sure what that means. But I know a lot of people are, and would see that as kind of a source of contingency. I would see God ordaining the contingency, but in a way that is often a bit mysterious. Yeah.
I wanted to have a follow-up question probably on this. When you talked about the extinction on the level of proteins, I was quite interested about whether there is a tendency to protect some of those or prevent some of those extinctions. When we see human behavior in the nature, we try to protect some species which are extincting and which are endangered. So is there a level of protection on the level of proteins as a tendency from scientists?
I'm struggling to find something to say to that. I'm kind of stumped. So I'm not quite sure what the role of scientists would be in protection there.
Do you mean at the cellular level? So within the cells, there's a very complex machinery to help proteins stay folded and functional and deal with them if they misfold.
Yeah.
Chaperones, proteasomes.
Yeah. I don't have more to say about the role of scientists there, but yeah.
I think our microphone holder has answered the question. It's her professional opinion. So we've got a couple of quite technical questions online before we go to one more in the room. Isn't adaptationism, you might want to read it, Zachary, while I read it out. Isn't adaptationism a subset of structuralism? Should we not think that a structure we observe is a result of previous necessary adaptation of an organism without which it would not have survived last? Does it mean that structuralists bear the burden of proof to show that adaptation isn't necessary for sustaining life?
So one comment in relation to that is I think Darwin saw himself in line with the people like William Whewell and others who were giving a law-based explanation of nature. But it's just natural selection is a very strange kind of law, because it's really just filtering by the environment, and the environment goes in all possible directions. So if adaptationism, so it's not really directly answering the question, but if adaptationism is a form of structuralism, it's quite a strange form. And I think they're better seen as kind of separate, where adaptationism is focused on the function for a particular environment, where structuralism is focused on the overall plan of the organism or the biophysical laws that give it a particular shape rather than another shape. And that's quite different to something which is focusing on the specific function in a specific context.
Thank you. Let's have one last question.
Thank you for your talk. Yes, it's a bit of a general question. I'd like to hear you talk a little bit more about teleology, and very helpful distinction between adaptionism as kind of one way of viewing the teleological question, structuralism as another. Do you think there could be a synthesis of the two? I guess I'm interested in how evolution changes our understanding of teleology. I guess I just was keen to hear you speak a little bit more.
Yeah, so my thinking on that is so let's say it's true that there are underlying laws that really govern evolution in some way, such as various biases. That can start to look a bit like functionalism or adaptationism if it turns out those laws are biased in a way that is useful. So bias isn't necessarily useful. The fact there is bias is, I would say, un-Darwinian, and just that by itself is going back to these pre-Darwinian views. But if it turns out these biases are actually useful, they really facilitate evolution, then that's kind of an added step I didn't really push for in this talk. Though I do think that's probably has to be the case, that for evolution to be so successful, I think it has to have not just biases, but useful biases. And then that's kind of a way to merge structuralism with something like functionalism or adaptationism, because there's a direction towards a useful goal.
Thank you. You've answered that so briefly. We've another technical question just to end with. Someone is asking, simple versus complex at the molecular level. Can a connection be made to mathematical or computational ideas of complexity? For example, do they contain less information in some way?
Yes, in a word. And the work of Ardern is really focused in this area, I'd say. And he's really approaching it in terms of what's called algorithmic information theory, which is a formal way to quantify simplicity. So yeah, do go to his papers for that.
Great. Thank you. So one last question.
Could you say a bit more about convergent evolution to complex structures? You gave the example of flagellae.
Yeah.
So having in bacteria and archaea having completely separate origins, but then ending up in the same complex structure.
Yeah.
And then some people would say that's so complex, how could it arise by evolution? Can you say a bit more about that?
Yeah, so I think it's important to understand why we think they have evolved. And that's what most people who have that kind of response don't fully understand, is why it's not just, we're not just pulling it out of the air that these things evolved. It's not just a atheistic presupposition that we have, but it's actually based on inference in the same way that we infer that English and German and various other more distant European languages are related. It's the same inference method that's used to infer that proteins are related to each other. And when we apply these methods, we see that there are relatives for the proteins. Now, we don't fully understand the evolution of the flagellum. There are some interesting pointers I could point people to, but we don't fully understand that. But we do see it in a context where there's lots of evidence for these proteins evolving. There's not just one flagellum. Each bacterial phylum has its own different kind of flagellum. So often this is talked about in a way that's unhelpful. Like, how did the bacterial flagellum evolve? Like, the first question will be which flagellum, and why are there so many, and why are they related to each other? So that's part of the context I need to talk through if someone's skeptical of them having evolved. But given the context of deep time and the relationship between proteins, it's a natural inference to think that there is an evolutionary ancestor for these things.
Thank you very much. So we've worked you extremely hard. Thank you very much for helping us to understand this today. Thank you. Don't forget to sign up for the screening of a documentary on artificial intelligence called The End of Humanity, and a panel discussion at 6 p.m. on Thursday, the 14th of November at Emmanuel College in Cambridge.